presentation. Thank you so much.
Okay, can you see my screen?
Yes, yes, we can see your presentation.
Here we go. Okay, thank you very much for that introduction. Yes, I teach at Cornell University Department of Microbiology. I also do research and collaborate with Department of Cell and Systems Biology at the University of Toronto. So first, I'm going to talk a little bit about vaccines and plants. Why? What are vaccines and plants? Well, the idea is to produce vaccine proteins, which can in plants which are routinely eaten, such as potatoes, tomatoes, bananas, and we have found that vaccine proteins, which are expressed in these plant tissues, can be consumed by humans and elicit a robust mucosal immune response. So, why would we bother to do something like this? Well, the World Health Organization estimates that about 4 million people, the vast majority of these being children under the age of 5, die every year from preventable infectious diseases. And traditional vaccines can be very expensive and hard to come by in some of these developing countries. And by making our vaccines and plants, we circumvent the need for syringes, refrigeration, or trained medical personnel. And all of these are major stumbling blocks to develop providing vaccines to the poor. So the pictures in this slide come from a colleague at Doctors Without Borders, and it just shows that plants can enhance the availability of inexpensive pharmaceuticals, including antibodies, therapeutic agents, and vaccines, to those in developing countries. So plants have two advantages for vaccines uh, uh, as a production platform. Number one, that you just consume the plant itself, so it acts as the delivery vehicle. And number two, the plant cells have a fixed cell wall. So if a protein is being expressed in the plant, it actually is protected from the harsh environment of the gastrointestinal tract, so that there's more of a chance that a lot more protein will be exposed to the immune surveillance system, and you'll get a stronger, a more robust immune response. And this is exactly what happens if you compare a plant-made vaccine versus an oral uh, conventional vaccine. So there's a number of applications for making vaccines in uh, plants, and the first ones that were developed were targeting developing countries, and they were the major killers of children that were focused on. So first was cholera, rotavirus, Norwalk virus. And then we started moving forward to more pandemic type diseases or global health issues such as influenza virus and human papillomavirus vaccines. And then most recently, plants have been used to make vaccines for One Health diseases, including Ebola, chikungunya, Rift Valley fever virus, and Middle Eastern respiratory syndrome virus. And here's some photos of a colleague of mine, Dr. Julian Ma at St. George's University of London. He produced monoclonal antibodies to block HIV. He produced them in tobacco plants, and he was able to make seeds from these plants and send them to sub-Saharan Africa, where HIV is still a problem. So this just gives you an idea of how easy it is to work with this sort of system. In case this still sounds very much like science fiction to you, there are companies that produce pharmaceutical proteins in plants, and the first one is known as Protalix. It's actually an Israeli company, and it produces a therapeutic agent known as glucosuberosidase, and this is an enzyme that treats Gaucher disease, and it's being produced in these huge bags on the left-hand side. These are 10-foot tall bags of carrot suspension cells. So you could actually drink the carrot juice and be provided with a daily requirement of this enzyme if you're lacking it, if it's have this genetic defect. And to make the glucocerebrosidase conventionally, it costs about $100,000 a patient a year. But this way, it's pennies a glass, so it becomes quite affordable, quite suitable, and easy for a patient to self-administer. Another example where plant-made vaccines have hit the news is with an Ebola outbreak that took place, I think, back in 2012. In this case, a plant virus vector, which we developed at Cornell University, was used to produce monoclonal antibodies towards Ebola, and a cocktail was made with using our vector and another vector based on tobacco mosaic virus. And this monoclonal antibody was provided to Americans who had contracted the disease while visiting West Africa during this outbreak, and seems to have worked quite well. So this is just another example where a plant-made vaccine can rise to the occasion and be rapidly upscaled and used for helping out in public health emergencies. So I hope I've convinced you so far that plant-made vaccines can provide a unique opportunity to make an impact on global health. So we could either purify the drug from the plant that we've produced in plants, which I'm showing with the corn here producing pills. So we can do that just our usual way and then provide those to developing countries. Or on the bottom here, I'm showing tomatoes. We can actually produce the drug or vaccine in tomatoes and just lyophilize, partially purify, because this is a food, crop. We don't have to go through the full purification. We can, after we lyophilize the tomato into a powder, we can resuspend it into a tomato juice, which can be provided to folks in developing countries. Or my favorite application of all is just provide the technology and the know-how to the scientific communities in developing countries so that they can develop their own pharmaceutical industry that focuses on uh, in diseases of their own, localities that are often ignored by the global north. Now, a few years ago, I became involved in a research on Middle Eastern respiratory syndrome. This was because I was contacted by several Egyptian scientists. And you might remember that MERS is in fact a coronavirus, just like COVID-19. And so this particular virus is carried by camels along ancient trading routes. And I've put up these maps just to give you an idea of where camels are in the world. And you can see that these camel trading routes and the camel culture of people owning camels and living with camels is very extensive. If you look along the map on the top right, you can see that probably 2 to 3 billion people of our world population are affiliated with camels. It's really quite remarkable. And camels have this endemic MERS virus, which is asymptomatic for them, but causes havoc for humans of 50% mortality rate. So I got involved in a project to make a antiviral protein in plants for Middle Eastern respiratory syndrome. And so here I actually traveled to Egypt, and I'm showing you that even in somebody's backyard there's camels, so they're just everywhere. They're almost pets for a lot of people. And at University of Toronto, a colleague made what he calls a ubiquitin variant. And on the bottom left, you can see a purple and gray, two purple and gray molecules. So the purple is our protein that binds very tightly to deubiquitinase, which is an enzyme produced by the MERS coronavirus. It binds tightly and outcompetes any other molecule that would bind to the deubiquitinase. As a result, the virus is stopped in its tracks. So this ubiquitin variant is synthetically made, and it's just, being optimized to bind very tightly and outcompete and basically, kill the virus. And so the plan is what I've been working on is to produce this ubiquitin variant on the surface of plant virus particles so that they can be provided as um, as an inhaler, as an aerosol to folks who either come down with MERS or who are or family members of someone who has potentially had MERS in their in the area. And of course, we've had the COVID pandemic, and so my colleague has actually made a ubiquitin variant to COVID-19 and to SARS and to MERS. So I have all three that I'm making in plant virus particles. They'll be expressed on the surface of these virus particles, and they can block infection by these types of viruses. So now I'm just switching a little bit to talk about cancer immunotherapy uh, using plant virus nanoparticles. So this has been a really interesting move for plant virologists, but we found that empty plant virus particles, so they're virus-like particles without any nucleic acid inside, they can't replicate, they're just shells, basically empty shells. If they're injected into a tumor in an animal model, they will reduce the progression of the tumor and even get rid of the tumor entirely. And so on the right hand side, you can see that the top is, I think it's a liver of animal that had a tumor and had cancer, and the virus has been added, these empty virus-like particles, and the tumor is regressive. On the bottom, it's a tumor that's just been injected with phosphate buffer saline, so it's negative control. So it's really amazing that a plant virus can do this, and it's not clear to us exactly how it works. The general theory right now is that the plant virus will enter the tumor and will somehow awaken the quiescent neutrophils that are available. So tumors make a microenvironment which shuts down or quiets down the immune system. The neutrophils are inactivated and in a regular tumor microenvironment, but somehow these plant virus nanoparticles will reactivate it. The neutrophils will, once activated, will secrete chemokines and bring in the rest of the immune cells to lyse the tumor. So it's really fascinating that this will work. And the great thing about it is uh, plant virus uh, uh, plant virus particles are very stable at room temperature and very inexpensive to make. So it gives a new option for developing countries, again, I think, to address cancers. So since plant viruses do this, this is one type of plant virus I'm showing you here. That's a rod-shaped virus. We know an awful lot about plant viruses just from our experiences in the field. We know what their coat proteins look like. We know the X-ray crystal structure of the coat proteins. We know exactly what the structure of these viruses look like. So we're able to very easily use these as carriers for drugs or for epitopes or for whatever. So we can actually make good use of these plant virus-like particles as payloads. So this is an example here. This is a tobacco mosaic virus. We can actually add an anti-cancer drug into the interior of this virus. So normally the virus would have its nucleic acid, which it's an RNA virus. So the RNA would be in the inner channel of this assembled virus-like particle. But we can get the virus to assemble without its RNA genome, and then in place of its genome, we can add an anti-cancer drug, and we can get this to go right into a tumor or some type of cancer and focus the drug in that region. And this figure just shows that we can use plant viruses. We can either add a fluoromer, something that will glow or light up, and use this to target. So it's more as a diagnostic. We can target where a tumor is. We can also add some kind of anti-cancer drug and load that up and use that as a therapy. So these plant viruses can be used both as diagnostics and as therapeutic agents. So some of the work again that I've done at University of Toronto uh, involves using a rod-shaped plant virus known as a potexvirus to uh, um, produce it as an anti-cancer therapy. So here I'm showing you on the top, it's the genomic organization of the plant virus, and the coat protein is the one we're interested in. So I actually was able to put the coat protein gene into a very uh, uh, well-known plasmid that most labs in developing countries I find seem to have. It's PET29, and it grows in E. coli, and it's a protein expression vector. So I can get people in labs that have you know, very low levels of sophistication to still work on projects with me. And so we can get using this plasmid and using not even any plants at all. We're just putting the plant virus coprotein into E. coli cells, BL21 cells. They'll assemble into virus-like particles in the E. coli. So here I'm just showing you the coprotein being expressed in E. coli, and we have on the left-hand side of this electron micrograph is wild-type virus. On the right-hand side, we have the coat protein assembling into these virus-like particles. They're shorter, but they work nonetheless. They're immunogenic, and they're helpful. So it's useful for me to collaborate with other scientists and help them get on their way, even if they don't know much about plants. This is just a close-up of what's going on with these potexvirus nanoparticles. And here I'm showing that we can actually conjugate drugs such as doxorubicin, which is an anti-cancer drug. We can conjugate that to the surface of these virus particles. We know exactly where it's going because we can conjugate it through a specific amino acid bond. And by loading either an antigen or a drug to the exterior loop of the capsid protein, we're able to use it as a really great vehicle to address a number of diseases. And future experiments, a lot of the work has been done in cell culture and animal models, and I don't believe anyone has yet started a clinical trial with humans, but that's just a matter of time before we start working in this direction. So my conclusions are that plant-made vaccines could slow the spread of major diseases and even some cancers, and they're inexpensive to produce. They do not require sophisticated equipment. They could even be produced locally to address the specific needs of a geographical region. And thank you very much for listening. Ready to take any questions.